Hello, everybody. Today we're just going to talk about DNA replication. It's a complicated topic, and I will have a couple of lectures on it. So let's just begin with this paragraph that we see in front of us. DNA replication starts at a specific sequence called the origin. So it's part of the actual DNA that certain enzymes will bind on to. So that first enzyme that binds on is DNA helicase. So it binds onto the origin, then it unwinds it. It unwinds it by breaking the hydrogen bonds and kind of like unzips it. And that results in something called the replication fork. So it's split like a fork. Because it was twisted before, there's going to be a lot of tension, and DNA topoisomerases, an example is gyrase, will join on and helps to relieve that tension. You've got these hydrogen bonds that are still open. So single-stranded binding proteins, SSBs, they will join on, really, really small pieces of protein that just binds on to keep it from joining back up again. So replication actually occurs in two different directions from the origin, and that's called a replication bubble. Now, of course, DNA molecules are very, very, very long, and so it would take forever, it would seem, to get this all done. So they actually have these replication bubbles all along the long molecule of DNA. So this is actually being replicated in many places at the same time. And because of that, it can only take about an hour to do the whole chromosome. So this diagram is a great diagram. You can see here's the original piece of DNA. It's in the helix form. Helicase joins on. The helicase breaks the hydrogen bonds, so it unzips it. And you can see here's one side and here's the other side. Topoisomerase, like a gyrase, joins on just to keep the tension, keeps it straight. And again, we have single-stranded binding proteins, which help to keep the strands from joining back up again so that the hydrogen bonds don't reform. So there are actually two sides to it. One's called the leading strand, and the other one is called the lagging strand. The difference is that the leading strand is assembled towards the origin, so it's always going towards where the replication fork is. The lagging strand is actually assembled away from the origin, and it takes longer. The leading strand, what happens first is that we have RNA primers that have to be added on. So let's go down to this note here. DNA can only be synthesized in the 5' to 3' direction. Free nucleotides are added onto the 3' end of the elongating strand. RNA primer, 10 to 60 base pairs, is annealed to the template strand by the enzyme primase. The primer is later removed. So primer has to be put on. DNA polymerase III adds complementary bases at about 50 nucleotides per second in humans to the elongating strand from floating freely in the nucleoplasm. So building this leading strand. So the leading strand is built heading towards the replication fork. It's the only strand that's assembled continuously. Like I said, there's the leading strand that's made continuously. Lagging strand, RNA primers are first added to sections of the template as it's opened from the replication fork. That's the first thing that happens. The lagging strand is assembled discontinuously away from the replication fork in short sections of 100 to 200 nucleotides. And those short sections are called Okazaki fragments. DNA polymerase I replaces the RNA primer between the Okazaki fragments with complementary DNA nucleotides, and DNA ligase joins the fragments together. Finishing the replication, the double-stranded DNA molecule automatically twists. The sequence is checked for mistakes by DNA polymerase I and II, and mistakes are removed. Well, I think learning always happens from replication, repeating. So we're going to repeat